Good afternoon. So today we will be talking about Process Analytical Technologies, and this abbreviation PAT is commonly used in this context. So what we discuss here today is first we have to think about the aspects related to sampling. So with process analytics, you try to understand something happening in the process. So then it's important to connect it to the sampling theories. And then I will talk about the basic principles of process analytics with a few process analytical examples, and then a bit more complex process analytics with the process spectroscopic tools. So this is the flow for today. So if you think about samplings, so you can take a classical analytical tools that are used in the laboratory. So this is something that is commonly done analysis for pharmaceutical materials. So this is a thermal analysis. And here you can see a small sample holder. So we can try to put the laser pointer on. So here in the middle, we have a sample holder where we have a 10 milligrams of sample in there. And then that is then used for understanding how much water there is in this sample. So you can do thermogravimetry and then measure all these particles and then they get an estimate of water content in here. So this is fine and most likely describes well this small sample here. But if you then try to link it to the bigger amounts of material there, then you need to do some considerations. And then if you, for example, are analyzing crystallization on API and your batch size is 1400 kilos, so then this one sample might be a bad representation of the total population. So you need to always think about the scale. So how to sample, how to measure optimally from the material. And especially this link here is challenging. So many times these materials, they are handled in a bigger amount. So you might have this type of big pack of material, and then you need to select the sample. And this is a bit challenging if you use offline analytical tools. So if you use, for example, this classical sample thief approach. So with sample thief, you have a metallic device that you just place in a powder and then rotate it, and then there will be opening few holes in there. And then what will happen is that you can collect from different heights of this powder bed and then different parts of it. And then by this means, by increasing the number of samples, then you increase the likelihood of getting a representative overall sample. Another approach is that you use sample divider. So here we have a sample divider that is rotating here, and then you pour your powder in here, and then while this is rotating, then you divide your original few kilos sample into smaller amounts. And by this means, then you increase the likelihood of getting a representative sample. But there's still this challenge of when you start with 1400 kilos and then your analytical method is able to handle 20 milligrams, then it is a really, really demanding task. And then you have to be careful on when you estimate the analytical method performance. And many times it's important to remember that your analytical method, if it gives you very precise results with the parts of percent and all this, so then you have to always remember that you have to include in the error all possible sources of error. And then this starts by thinking about the primary sampling of your material. So you do the primary sampling on the site where you are producing the API or an excipient. So then you have to think about what is the total size of the sample that you have, the total size of the material that you have, and then how would you collect the representative sample of that. And then this sampling usually contains two steps. So primary sampling you indeed do at the production site. So you try to get from this big container a few grams of material in a representative way. And then you take that sample typically to a more clean environment. So you take it to your laboratory and then you do secondary sampling. So then you have that few grams of material and then you divide it into smaller parts so that you finally will have your 20 milligram sample. So this is then producing you the total analytical error. So you need to include all these aspects into the estimation of the overall error. And we often talk about primary sampling error and then secondary sampling error. And then when you sum up these, then you will have the global estimation error. And it's important to remember that this analytical error is only part of the total estimation error. So if you are saying that your tablets contain 100.007 milligrams of active compound, so then you have to be a bit careful in there with the decimals, so that you include also the global estimation error in the total estimation. And this is something that has been quite nicely presented by Pierre Gu, that is a person focusing on sampling errors, and that's a quite theoretical considerations in there. But this is something that is kind of a good bridge for thinking about process analytics. So with process analytical methods, we are trying to move the analytical methods to from laboratory towards the production environment. So the idea is that you would do the analytical measurement in the production environment. So, and then with this means, then you can reduce this global estimation error. And instead of doing HPLC analysis of 10 tablets, you could consider doing process spectroscopic measurement in the site of production and from 100,000 tablets. So by this means, then you reduce the overall estimation error and then can get the better analytical result. So you are always contrasting with the, so to say, offline laboratory work when you are doing process analytics. Yes, so this was a kind of a warming up for this process analytical discussion. And then in the next part of the lecture, I would like to show you a few examples of process analytics. And this is something that is related to risk-based product development. So the idea of risk-based product development, or quality by design, QPD, if you will, is based on the fact that you should start the work always by doing proper risk assessment of your product. And this starts by doing quality target product profile, QTPP, where you structure the elements of the product that you want to build in there. And then those elements, they can be described with the critical quality attributes. For example, for an immediate release tablet, this could be the disintegration time. That would be a critical quality attribute. And in order to understand your system, so your risk assessment is giving you the most important elements to focus on, and those you can then investigate by doing structured experimental design, usually using design of experiments tools. And then what you could consider doing, for example, for the immediate release tablet, is that you investigate the effects of different granulation parameters and then the composition of the tablet on the disintegration. And then that you can do with the design of experiments. And once you have nailed down the factors that affect the system, and then you know how they affect it, then you can start thinking about implementing some process analytical tools for controlling them. And the idea of PAT is that you have an idea how variation in the system affects the quality of your product. And then you build up process analytical solutions that you can measure during processing what is happening, and then do corrective actions. So change the process parameters so that you get always the variation, minimal variation that you want for the product. And I cannot give you a single solution for all possible pharmaceutical processes. So it's important to remember that we are dealing with many different types of processes. And then depending on the process, then you might need a slightly different analytical tools. So if you are producing biopharmaceuticals, protein peptide-based products, so these are typically done by fermentation. So you have a cell line that is expressing the product and the active compound, and then you separate it and purify it with the chromatographic processes. So this is fundamentally different from small molecule system, where you typically have organic synthesis that is producing the active compound, and which then is purified by crystallization and then downstream processed with granulation, tabletting, or coating. So for these processes, all processes can be analyzed with the different process analytical tools, and then there are hundreds of different PAT tools out there, and I plan to show you a few examples how this can be done. My examples, they are from the small molecule side, but then you have to just extrapolate in your mind and then think about doing the similar analysis on the protein world if that is of interest for you. So if we look through this flow diagram of processes typically involved in a small molecule side. So we start with this synthetic reaction, and then via crystallization, granulation, mixing processes, then we proceed to tabletting and coating. So these are typically done sequentially, and quite often the analytical burden is at the end of this line. So we sample from coated tablets and then try to make sure that our product is meeting the specification. And it's very unfortunate that if you find out from the coated tablet that something started going wrong already in the synthesis. So that is another driver for doing process analytics is that you can already do early detection of possible problems in the product. So you can identify already in the synthesis that something is going wrong. That then most likely accumulates during processing, and then synthetic impurity is increasing. And then finally you find out from the coated tablet that it's containing too much impurities. So the idea would be that you implement these processing steps with different analytical tools, and then always produce the desired quality. And there are many different tools out there. So we can use spectroscopic tools. I will use a bit more time on discussing about these. But then similarly, you can do process analytics with, for example, image analysis or different diffraction tools, or you can measure mechanical properties of material during processing. The good example of that is a tabletting process, where you can measure the force needed to make a tablet, and that would be a mechanical response of processing. You can also measure thermal properties. Simply the temperature of the process can tell many things. And you can even collect acoustic or electrostatic information from process. So you can measure acoustic signal of tablets and then proceed and then, for example, investigate the compaction process or coating processes when the acoustic properties of material and then the container walls are changing. So this is many different possibilities out there, and then there are many things that I cannot here in this short lecture cover, but these are a few examples that would be useful for understanding pharmaceutical processes. And whenever you decide to pick up a process analytical tool, then it's important to think about the physics of the measurements. So here we can use a simple temperature measurement as an example. So we often talk about probes. So for example, temperature probe is something that you use for measuring temperature from the moving material. And commonly used solution is a resistance temperature detector. And then these are typically consisting of this type of metallic wire. And then you calibrate the resistance of this one against the known temperature. So like a ice water and then boiling water or some other temperature standard. So then you build up a calibration curve for the resistance of this one, and then you make sure that you always measure the very precise temperature of a system. And then this system is of course covered with a robust stainless steel, so that you can place this instrument in contact with a synthetic reaction medium or crystallization medium or even granulation medium, and then always measure the temperature of the material during processing. And this is important for understanding many complex things. So in the same way, whatever you want to measure, so always start by thinking about the physical principle of this measurement, and then make sure that you calibrate your instrument for its intended purpose. just to give you an idea what this can mean and then how our user will see this one. So this is an example of fluidized bed granulation. It is a commonly used process in the pharmaceutical field. And what we have here is a user interface. So user interface is used by the process operator to look into different measurements during processing. So here in the lower part of the fluid bed granulator, we have the powder material. So you have a few kilos of material here, depending on the scale of the instrument, or 100 kilos of material. And then you have the air coming in from the bottom, and that air is used to fluidize the powder. And then you add the granulation liquid from top. So then you can increase the granule size. So important parameters here in this process are the flow rates of air, temperature of the air, and then the granule moisture content. And then this can be measured with many different approaches. So here we have an example of instrumentation of this process. So each spherical symbol here is representing one temperature probe. So here we can see these green symbols that are referring to temperature measurement. Then we can also measure the flow rates of air. We can measure the relative humidity, absolute humidity of air that is going in. And then in the same way, we can measure also the properties of air when it's going out from the process. So here on the top of the granulation equipment, we have filters, and then we can measure again the temperature and then relative humidity of the outgoing air. And these measurements can be used for building up a mass balance around this granulation process. So then we can investigate how much water is in granules, and then by this means we can control the moisture content of granules and during the granulation, and then also while we are drying them. And typically one granulation will look something like this here in this corner of the user interface. So what we can see here is a time series of these measurements, and then we can see that during granulation we have first a drop in the granule temperature, and then a slight increase in it, and then some fluctuation, and then also some decrease at the end of the process. So this is describing the state of the granules during processing. So this pressure difference and temperature difference between granules and the incoming air can be used to describe how much water there is at the surface of the granule. And that is then related to different granule growth mechanisms and aspects like that. So these measurements can be used for understanding what is happening in the process. And this is also a unique fingerprint of your process. So you can see that you have always done it according to the expected temperatures and expected flow rates, so that there are no big hiccups there in the process. So this is something that is very useful for understanding your process. And you can take each measurement one by one and then start thinking that what is happening, for example, in this flow rate, and what is happening for the temperature of incoming air. And then we can look some different temperature differences in the process, and then some differences in the water content of the samples, and then some pressure differences. And then even here in this setup, we have a direct measurement of water content with the NIR probe. So this is all presented as a function of processing time. And then this gives you a unique fingerprint for understanding this granulation process. So in this specific case, we can see that maybe the most clear signal is the water content. So what happens here is that in the granulation process, you start by mixing the powder, and then you add the granulation liquid, and then you can see that the water content is increasing. And then when you are doing the drying phase, then the water content is decreasing. So this is a fingerprint of your granulation cycle. And then you can see how different other measurements are behaving while you are doing that. So what we can see here is that we are, for example, changing the temperature of the incoming air when we are having the drying phase. And then that is then related to many other measurements. So it can speed up the drying process. And then by this means, then you also see some changes, for example, in the pressure difference over filters, for example. So by this means you can measure many different parameters at the same time, and that gives you an overview of this specific process. And one important aspect of doing these process analytical activities is that you implement also process control aspects into the work. So here we have a schematic illustration of what process control means. So here we can imagine the controlled variable that can be measured, for example, temperature of the air in the process, and then you have a measurement of that, and then you have a set point of that value. So you have the variable that you want to control, so you say that it needs to be 60 degrees, and then this measurement is saying that it's only 57. So then there's a difference, and the idea with this feedback control is that you measure the difference between set point and measurement, and then based on that, then you manipulate something before the process. So for example, here the manipulated variable can be the heating element in the incoming air. So if you know that there's a 3 degrees difference, then you need to increase the energy in the heating element, and then increase the temperature of that so that you can get the desired temperature. And then outcome of this is typically that you have this type of a cycling measurement of your around your set point. So if you want the temperature to be 40 degrees here and 60 degrees here, so then typically these control loops will fluctuate around the set point value. So this is quite understandable. So if you heat a metallic element and then turn off the heat, so then the temperature will fluctuate. So typically an expert within pharmaceutical sciences don't end up being the person tuning these controllers, but it's important to consider what is the consequence of this type of fluctuation. So we cannot build up perfect control loops that would have no fluctuation. So there's always this fluctuation, but you need to estimate that this process measurement is having acceptable variation in the around the set point. Yes. And I think this might be the point where we take a short break and continue after a while.